Welcome to Science Education Channel. In this video we will be discussing about spoilage of food 1. Classification of food on the basis of ease of spoilage 2. Sources of microorganisms in food. Microbial load 3. Spoilage of vegetables, factors responsible and types of spoilage. 4. Spoilage of meat and meat products, factors responsible and types of spoilage. 5. Spoilage of canned food, factors responsible and types. 1. Spoilage of food. Definition of food. Microbiology, a branch of microbiology which deals with the study of microorganisms associated with the food and food products. Food spoilage, food poisoning, food infections and the preservation of food and food products is known as food microbiology. Food microbiology concerned with the following 3 objectives. 1. Prevention of food contamination and the preservation of foods and food products to eliminate microorganisms responsible for food spoilage. 2. The detection and prevention of food poisoning and food infections due to pathogenic microorganisms. 3. Application of microorganisms to manufacture new nutritive foods and to improve desirable characters of the food. Types of food. Human food supply consists basically of plants and animals or products derived from them. Types and kinds of food vary from country to country and place to place. However, foods that are commonly eaten all over the world can be classified into 2 main types. A. Food derived from plants. B. Food derived from animals. A. Food derived from plants. X. 1. Circle cereal products. 2. Sugar and sugar products. 3. Vegetable and vegetable products. 4. Fruits and fruit products. B. Foods derived from animals. X. 1. Milk, meat and meat products. 3. Poultry and eggs. 4. Fish. Classification of food on the basis of ease of spoilage. On the basis of ease of spoilage, foods can be placed in 3 groups. I. Non-perishable or stable foods. These foods are not spoiled easily by microorganisms unless handled carelessly. X. Cereals, grains, rice, wheat, flour, nuts, sugar etc. E. Perishable or unstable foods. These foods are spoiled by microorganisms very easily due to their high nutritive value. X. Vegetables, fruits, eggs, milk and milk products, meat, fish, poultry etc. E. Semi-perishable foods. These foods are spoiled comparatively less quickly. If properly handled and stored, they can remain unspoiled for fairly long period. X. Potato, onion, garlic, apples, nutmeg, spices etc. Based on their physical state, common food items can be classified as 1. Fresh foods. 2. Preserved and stored foods. 3. Canned foods and cured foods. 4. Processed foods. 5. Fermented food products. 6. Cooked foods. To be safe for human consumption, food must be free from harmful microorganisms. Herefore, food and food products must be carefully prepared, processed and stored. Microbial food spoilage. Introduction. All types of food items are associated with the microorganisms. Most of the food items functions as an excellent nutrient media for the growth of many microorganisms which are mostly saprophytic bacteria, actinomycetes and fungi. Food and food products gets contaminated with microorganisms in any one or more one of the following ways. Sources. 1. From plants, soil, water and sewage. 2. During harvesting. Transport and storage. 3. During collection and handling. 4. During cooking and preparations. 5. From utensils in which they are prepared and stored. Microbial growth on foods causes its deterioration or decomposition, changes its flavor, texture and nutritive value resulting into the food spoilage. Food spoilage makes the food unsafe for consumption. Some pathogenic microorganisms cause food infections, ex. Salmonellosis, Shigellosis, cholera etc., whereas some produce toxins and cause food poisoning, e.g. botulism, staphylococcal poisoning. Food spoilage by microorganism depends upon the physical and chemical properties of the foods. The process by which the food is made useless and unsafe for eating is called food spoilage. Foods and food products are contaminated by various sources such as plants, animals, soil, air, water, sewage, unhygienic habits of food handler, processing, utensils etc. Most foods serve as ideal media for the growth of many different microorganisms. Contaminated microorganisms grow and multiply in the foods and changes, physicochemical properties. Appearance, texture, color, taste, flavor, odor, stability and nutritive value and make the food unsafe for consumption. Such undesirable alterations or changes brought about by microorganisms in the food and food products are called microbial food spoilage. Food spoilage may be due to one or more of the following reasons. 1. Growth and activity of microorganisms or plant pathogens or insects. Action of native enzymes in the plant and animal foods. Certain altered chemical reactions in the food. Some physical changes occurring in the foods during freezing, burning, drying, processing etc. Factors responsible for food spoilage. Some of the important factors responsible for food spoilage by microorganisms are 1. Intrinsic factors, 2. Extrinsic factors, 3. Chemical composition of food. 1. Intrinsic factors are those that are maturely present in the plant and animal tissues from which we obtain the food. This includes pH, moisture content, redox potential, nutrient content, antimicrobial constituents etc. 2. Extrinsic factors are those which are present in the environment where foods are kept and stored. These includes microbial population of the environment, moisture content and pH of the environment, storage, temperature, condition of raw products, the method of handling, the time and conditions of storage, the type and concentration and gases in the environment etc. The inner tissues in healthy plants and animals are free of microorganisms. However, the surfaces of raw vegetables, fruits, cereals, fish, meat, poultry etc. are contaminated with a variety of microorganisms. 3. Chemical composition of food. This factor play very important role in spoilage by microorganisms. Some foods are acidic in chemical nature, acidic foods, some basic in nature, basic foods, whereas some are neutral in nature, neutral foods. Further, most of the food and products are chemically composed of carbohydrates, proteins and fat, lipids. Depending upon the chemical composition, food spoilage by microorganisms may be caused by following biochemical reactions. 1. Putrefaction, decomposition of proteins. 2. Fermentation. 3. Rancidity, decomposition of carbohydrates. Decomposition of fat, lipids. 1. Putrefaction. The foods rich in proteins are usually decomposed and spoiled by anaerobic and facultative anaerobic microorganisms which produce protease enzyme resulting into putrefaction. Putrefaction of food leads to the production of foul-smelling compound, such as H, S, ammonia, mercaptans, amines etc. It is caused by Proteus, Pseudomonas, Clostridia, Leuconostoc, Rhizophis, Aspergillus etc. 2. Fermentation. The foods rich in carbohydrates are decomposed and spoiled by facultative anaerobic microorganism which produce amylase enzyme resulting into fermentation and production of ethanol and company. The partial fermentation of carbohydrates leads to the production of various organic acids such as acetic acid, lactic acid, butyric acid, formic acid etc. It is caused by Streptococci, Micrococci, E. coli, Saccharomyces, Acetobacter etc. 3. Rancidity. The foods containing fat, lipid are decomposed and spoiled by the microorganism which produce lipase enzyme, resulting into rancidity. Rancidity of food leads to rancid odor and taste in food. It is caused by facultative anaerobic microorganisms such as Pseudomonas, Bacilli, Achromobacter. Some food spoilage by microorganisms. Types of food spoilage and causative organisms. Foods type of spoilage microorganisms involved bread moldy ropey male sap and syrup ropey yeasty Rhizophus nigricans, Aspergillus niger. Bacillus subtilis Enterobacter aerugine Saccharomyces and Zygosaccharomyces pink Micrococcus rosis moldy Aspergillus and Penicillium species. Sources of microorganisms in food. The foods of plants and animal origin carry several microorganisms associated with their natural habitat. Plants carry typical microflora on their surface and also get contaminated from outside sources. Animals carry microorganisms on their surface and intestine, and also contain contaminants from surrounding environment. Through their excretions and secretions, animals release microorganisms into surrounding environment. Besides, both plants and animals carry pathogenic microorganisms capable of causing human illness. The food-associated microorganisms are influenced by the availability of specific nutritional requirements. And the environmental parameters. The primary sources of entry of microorganisms into foods are from the soil, water, air, during handling, processing, transportation and storage of foods. Soil. Soil, being the rich source of several kinds of microorganisms, immediately contaminates the plants and edible plant parts, and the surface of animals with the soil-associated microorganisms. As the soil particles are carried into aquatic environment through wind, rain and other means, contamination of water takes place with several soil microflora. Therefore, it is not uncommon to find several microorganisms both in soil and water environment. These soil-derived microorganisms form part of the microbial flora involved in spoilage of foods of plant and animal source. Thus, there is a need to reduce the load of soil microorganisms in foods, which can be achieved by removing the soil by washing the surface of foods with good quality water, and by avoiding contact with soil, dust water. Natural waters not only contain several microorganisms native to the aquatic environment but also from soil, raw, treated sewage and pollutants entering the water body. The microbial numbers and types vary in different water bodies depending on the nutrient status. Thus, all kinds of microorganisms found in water are likely to be associated with the aquatic organisms as surface flora. Use of such water for food processing will add microorganisms from water to food. Sewage waters containing human pathogenic microorganisms contaminate foods when such waters are used without proper treatment. The water used in food processing should meet agreeable chemical and bacteriological characteristics. Air. Air contains several microorganisms which may get deposited on the food being processed and handled. Though the air does not contain natural flora of microorganisms, whatever microorganisms encountered are those associated with the suspended solid material and water droplets. The sources of microorganisms to air are from dust, dry soil, and water spray from natural surface waters, droplets of moisture from coughing, sneezing and talking by food handlers, from sporulating molds growing on walls, ceilings, floor. Foods and food ingredients. Thus, it is likely that the microorganisms persisting in air get deposited on the food being processed and contribute for microbial load and subsequent spoilage of food. The number of microorganisms present in air depends on factors such as extent of movement of air, sunshine, humidity, location and amount of suspended dust in air. Quiet air allows settling of microorganisms, but the moving air brings in microorganisms and keeps them suspended. Thus, the number of microorganisms in air is increased by air currents caused by movement of people, by ventilation and by breeze. The rain or snow removes microorganisms from the air. During handling and processing, personnel in food processing plants can contaminate foods during handling and processing. Various workers suggest that human beings shed from 103 to 104 via-Bay organisms per minute. The number and types of organisms shed are closely related to the subject's working environment. Microbial spoilage of vegetables. Introduction. Vegetables form an integral part of diet due to their role in providing various types of vital nutrients such as carbohydrates, minerals, vitamins, roughage etc. Vegetables, being a part of fresh produce, contain high moisture, which makes them highly perishable foods and hence more prone to spoilage. Microorganisms gain entry into vegetables from various sources. These sources include soil, water, diseased plant, harvesting and processing equipments, handlers, packaging and packing material, contact with spoiled vegetables. The conditions in which vegetables are stored and transported after harvesting also contribute to rate of spoilage. Other than microbial sources, the spoilage of vegetables can also occur due to the activity of native enzymes. Types of spoilage in vegetables. The microbial spoilage of vegetables is predominantly of following types: spoilage due to pathogens. The plant pathogens which infect stem, leaves, roots, flowers and other parts or the fruit itself. Spoilage due to saprophytes. Vegetables have general microflora and spoilage due to saprophytes. Vegetables have general microflora inhabiting them. These organisms under certain conditions can grow on these vegetables and spoil them. The list of these organisms is given in Table 8.1. There are certain secondary invaders which may enter the healthy food or grow after growth of pathogens. It is well known that plant diseases are mostly caused by fungi. Thus, most of the spoilage causing pathogens in vegetables are fungi. Fungi have specific characteristics when spoiling food is it leads to mushy areas which may be water-soaked. The fungi produce characteristic spores which may be pigmented. The pigmentation helps in identification of the type of spoilage by fungi. The bacterial diseases too cause spoilage of vegetables, but to a lesser extent. Spoilage in vegetables is largely affected by composition of vegetable. The non-acidic foods are thus spoiled by bacterial rot, while acidic foods with dry surfaces are more prone to mold spoilage. The product on which organism grows and types of organisms growing largely determine the character of spoilage. Bacterial soft rot caused by Erwinia carotivora and Pseudomonas such as P. marginalis, Bacillus and Clostridium spp. Are also implicated. Breaks down pectin, giving rise to a soft, mushy consistency, sometimes a bad odor and water-soaked appearance. Vegetables affected: onions, garlic, beans, carrot, beets, lettuce, spinach, potatoes, cabbage, cauliflower, radishes, tomatoes, cucumbers, watermelons. Fungal spoilage of vegetables: Penicillium, Cladosporium, Rhizophis, Aspergillus spp. Are responsible for various defects in vegetables. Grey mold rot caused by Botrytis cinerea in vegetables. Favored by high humidity and warm temperature. Fungal spoilage of vegetables, Penicillium. Cladosporium, Rhizophis, Aspergillus spp. Are responsible for various defects in vegetables. Grey mold rot caused by Botrytis cinerea in vegetables. Favored by high humidity and warm temperature. Spoilage of meat. Meat contain comparatively more amount of moisture, proteinous compounds, nitrogenous compounds, minerals, vitamins, fermentable carbohydrates and favorable pH for the growth of many microorganisms. Therefore, meat spoilage is caused by many bacteria, molds and yeasts. Interior of the meat is spoiled by anaerobic bacteria, whereas aerobic microorganisms cause spoilage at the surface. The common type of meat spoilage is as follows. A. Meat spoilage under aerobic condition. 1. Spoilage by bacteria. The bacteria like Pseudomonas, alkaligenes, Leuconostoc, Klebsiella etc. grow on the meat surface and produce slime, resulting into gelatinous or slimy appearance of meat. B. The growth of Lactobacillus species on meat produce H2O2, which oxidize red-colored pigment of the meat to green color, called green meat spoilage. A. The growth of lipolytic bacteria such as Pseudomonas, Achromobacter, alkaligenes etc. degrades lipid, fat in the meat and produce rancid odor. D. Growth of certain pigment-producing bacteria cause discoloration of the meat. For X, Micrococcus species results yellow color. Pseudomonas species blue color, Serratia species red color. 2. Spoilage by molds. Various species of molds grow and produce spores on the meat, resulting into the fuzzy appearance, stickiness and discoloration of meat. Important molds causing meat spoilage are Penicillium, Aspergillus, Rhizophis, Cladosporium etc. 3. Spoilage by yeast. Certain yeast species grow on the meat and results rancidity, lipolysis, sliminess and discoloration of meat. B. Meat spoilage under anaerobic condition. Anaerobic bacteria usually grow interior of the meat and cause its spoilage. Anaerobic meat spoilage results following defects. I. Putrefaction. It is anaerobic decomposition of proteins in the meat with the production of foul-smelling compounds like amines, HS, mercaptans etc. It is caused by Clostridium species, Proteus species, Pseudomonas species, alkaligenes etc. E. Souring of meat. Certain facultative anaerobic bacteria decompose carbohydrates in the meat and produce various acids such as acetic, butyric, propionic, lactic, succinic etc., resulting into the souring of meat. It is caused by Lactobacillus, Acetobacter, Propionibacter, Micrococcus etc. Spoilage of canned foods. Canned foods are spoiled due to the entry of microorganisms through the leakage of container and contamination by endospore-forming microorganisms during processing, loading and storage. Spoilage of canned foods usually takes place by two types of bacteria. 6. Thermophilic bacteria, i.e., mesophilic bacterial. 1. Spoilage by thermophilic microorganism. The endospore-forming and thermophilic bacteria can resist canning temperature, 80 to 90 degrees Celsius, and grow in the canned foods, resulting into the various types of canned food spoilage as below. A. Flat sour spoilage. The growth of thermophilic, B. coagulants, B. Stereothermophilus cause production of acids and alcohols from carbohydrates and coagulation of proteins, resulting into unpleasant sour taste, abnormal odor and sometimes cloudy liquor in the food content of the can. This type of spoilage is not detected unless the can is opened. B. Can swelling spoilage. Growth of thermophilic anaerobic spore-forming Cl thermosaccharolyticum produce various acids and carbon dioxide H gases which cause swelling of the can. More accumulation of CO and H may result bursting of the can. The canned food becomes sour, gelatinous and develop butyrous odor. The growth of aerobic spore-forming bacillus species also cause swelling of the can, if the canned food is cured meat. C. Sulfide spoilage or black spoilage. Growth of Cl. nigricans and Proteus vulgaris produce H2S, which react with iron in the canned foods and develop black color. Such spoiled canned food gives rotten egg odor. E. Spoilage by mesophilic microorganisms. The bacteria which grow in the mesophilic temperature range, i.e. 25 to 40 degrees Celsius, optimum 37 degrees Celsius, results canned food spoilage. These bacteria enter into the canned foods through leakage of the container during cooling and handling. The mesophilic microorganisms which cause canned food spoilage are as follows: a. Growth of Cl. Butyricum and Cl. Pasteurianum produce butyric acid, also carbon dioxide and H gases, resulting into the swelling of the can. These bacteria usually grow in the acidic canned foods, such as tomato juice, fruit juices, jam, jellies, or medium acidic canned foods like corn, peas, spinach, beans etc. B. subtilis and B. mesenteroid brings about the spoilage of canned seafoods, fishes, and meat. A. B. polymixa, B. maceran, Streptococcus lactis, Pseudomonas species, Proteus species and some molds, e.g. Aspergillus. Cladosporium, also cause spoilage of canned foods. D. Growth of yeasts in canned foods produces alcohol and company, which results swelling of the can. Factors affecting spoilage, extrinsic and intrinsic factors in detail. Food is a chemically complex matrix, and predicting whether, or how fast, microorganisms will grow in any given food is difficult. Most foods contain sufficient nutrients to support microbial growth. Several factors encourage, prevent or limit the growth of microorganisms in foods. The most important are water activity. AO, pH and temperature. Factors affecting microbial growth are divided into two groups: intrinsic and extrinsic parameters. Table 2. These factors affect the growth of microorganisms on foods. When spoilage of a food occurs under a given set of circumstances, not all of the different types of organisms contaminating a food are associated with the spoilage process. In fact, the spoilage flora always is dominated by just a few and sometimes only one organism. Components of the microflora compete with one another for the available nutrients, and the organisms with fastest growth under a particular set of circumstances will become dominant and give rise to the spoilage symptoms. Intrinsic parameters: water activity, humectant identity, oxygen availability, pH, acidity, acidulant identity, buffering capacity, available nutrients, natural antimicrobial substances, presence and identity of natural microbial flora, colloidal form. Extrinsic parameters: temperature, relative humidity, atmospheric composition, packaging. The component of the microflora which becomes dominant is determined by a complex interaction between the components of the contaminating microflora, implicit factors, the storage environment, extrinsic factors, and the physico-chemical properties of the food, intrinsic factors. Extrinsic factors: Extrinsic factors relate to the environmental factors that affect the growth rate of microorganisms. They are as follows. A. Temperature. Microbes have an optimum temperature as well as minimum and maximum temperatures for growth. Therefore, the environmental temperature determines not only the proliferation rate but also the genera of microorganisms that will thrive and the extent of microbial activity that occurs. For example, a change of only a few degrees in temperature may favor the growth of entirely different organisms and result in a different type of food spoilage and food poisoning. These characteristics have been responsible for the use of temperature as a method of controlling microbial activity. The optimal temperature for the proliferation of most microorganisms is from 150 to 400 degrees Celsius. However, many genera of microbes are capable of growth from 00 to 150 degrees Celsius, and other even microorganisms will grow at sub-zero temperatures. Still other genera will grow at temperatures up to and exceeding 1000 degrees Celsius. Microbes classified according to temperature of optimal growth include thermophiles, high temperature loving microorganisms, with growth optima at temperatures above 450 degrees Celsius, e.g., Bacillus stearothermophilus, Bacillus coagulans, and Lactobacillus thermophilus. Mesophiles, medium temperature loving microorganisms, with growth optima between 200 and 450 degrees Celsius, e.g., most Lactobacilli and Staphylococci. Psychrotrophs, cold temperature tolerant microorganisms, which tolerate and thrive at temperatures below 200 degrees Celsius, e.g., Pseudomonas and Acinetobacter. Bacteria, molds and yeasts each have some genera with temperature optima in the range characteristic of thermophiles, mesophiles, and psychrotrophs. Molds and yeasts tend to be less thermophilic than bacteria. As temperature approaches 00 degrees C, fewer microorganisms can thrive and their proliferation is slower. As temperature falls below approximately 50 degrees Celsius, proliferation of spoilage microorganisms is retarded as the growth of nearly all pathogens ceases. B. Oxygen availability. As with temperature, the availability of oxygen determines which microorganisms will be active. Some microorganisms have an absolute requirement for oxygen, whereas others grow in total absence of oxygen. Yet other microorganisms can grow either with or without available oxygen. Microorganisms that require free oxygen are called aerobic microorganisms, e.g., Pseudomonas spp, and those that thrive in the absence of oxygen are called anaerobic microorganisms, e.g., Clostridium spp. Microorganisms that can grow with or without the presence of free oxygen are called facultative microorganisms, e.g. Lactobacillus spp. C. Relative humidity. This extrinsic factor affects microbial growth and can be affected by temperature. All microorganisms have high requirements for water to support their growth and activity. A high relative humidity can cause moisture condensation on food, equipment, walls, and ceilings. Condensation causes moist surfaces, which are conducive to microbial growth and spoilage. Also, microbial growth is inhibited by a low relative humidity. Microorganisms. Bacteria require the highest relative humidity of the various. Optimal relative humidity for bacteria is 92% or higher, whereas yeasts need 90% or higher, and for molds, the value of relative humidity is 85 to 90%. Intrinsic factors. Intrinsic factors that affect the rate of proliferation relate more to the characteristics of the substrates, foodstuff or debris, that support or affect growth of microorganisms. These major intrinsic factors are: A. Water activity, AO, water is required by microorganisms, and a reduction of water availability constitutes a method of food preservation through reduction of microbial proliferation. It is important to recognize that it is not the total amount of moisture present that determines the limit of microbial growth, but the amount of moisture which is readily available for metabolic activity of microbes. The unit of measurement for water requirement of microorganism is usually expressed as water activity, AW. Water activity is defined as the vapor pressure of the subject solution divided by the vapor pressure of the pure solvent, AOP, P0, where P is the vapor pressure of the solution and PO is the vapor pressure of pure water. The approximate optimal AO for the growth of many microorganisms is 0.99. And most microbes require an AO higher than 0.91 for growth. The relationship between relative humidity, RH, and AO is RH-AO 100. Therefore an AO of 0.95 is equivalent to an RH of 95%. Generally, bacteria have the highest water activity requirements of the microorganisms. Molds normally have the lowest AO requirements, with yeasts being intermediate. Most spoilage bacteria do not grow at an AO below 0.91, but molds and yeasts can grow at an AO of 0.80 or lower. Molds and yeasts are more likely to grow in partially dehydrated surfaces, including food, whereas bacterial growth is retarded. pH. The pH for optimal growth of most microorganisms is near neutrality, 7.0. Yeasts can grow in an acid environment, but grow best in an intermediate acid, 4.0-4.5, range. Molds tolerate a wider range of pH, 2.0-8.0, although their growth is generally greater with an acid pH. Molds can thrive in a medium that is too acid for either bacteria or yeasts. Bacterial growth is usually favored by near neutral pH values. However, acidophilic, acid-loving, bacteria will grow on food or debris down to a pH value of approximately 5.2. Below pH 5.2, microbial growth is dramatically reduced when compared from growth in the normal pH range. C. Oxidation reduction potential. The oxidation reduction, redox, potential is an indication of the oxidizing and reducing power of the substrate. To attain optimal growth, some microorganisms require reduced conditions while others need oxidized conditions. Thus, the importance of the oxidation reduction potential is apparent. Aerobic microorganisms grow more readily under a high oxidation reduction potential, oxidizing reactivity. A low potential, reducing reactivity, favors the growth of anaerobes. Facultative microorganisms are capable of growth under either condition. Microorganisms can alter the oxidation reduction potential of food to the extent that the activity of other microorganisms is restricted. For example, anaerobes can decrease the oxidation reduction potential to such a low level that the growth of aerobes can be inhibited. D. Nutrient requirements. In addition to water and oxygen, except for anaerobes, microorganisms have other nutrient requirements. Most microbes need external sources of nitrogen, energy, i.e., carbohydrates, proteins, or lipids, minerals, and vitamins to support their growth. Nitrogen is normally obtained from amino acids and other non-protein nitrogen sources. However, some microorganisms utilize peptides and proteins. Molds are the most effective in the utilization of proteins, complex carbohydrates, and lipids because they contain enzymes capable of hydrolyzing these molecules into less complex components. Many bacteria have a similar capability, but most yeasts require the simple forms of these compounds. Minerals, micronutrients, are needed by all microorganisms, but requirements for vitamins vary. Molds and some bacteria can synthesize enough B vitamins to fulfill their needs, while other microorganisms require a ready-made supply. Inhibitory substances. Microbial proliferation can be affected by the presence or absence of inhibitory substances. Substances or agents that inhibit microbial activity are called bacteriostats, and those which destroy microorganisms are called bactericides. Some bacteriostatic substances are added during food processing, i.e., nitrites. Most bactericides are utilized as a method of decontaminating foodstuffs or as a sanitizer for cleaned equipment.